Hello everybody, and in this screencast we're going to learn how to write a lab report. This is something that you will be doing not only in biology, in APES, in chemistry, but in physics, in AP biology, anatomy and physiology, and all of your other science courses. So this is the basic setup that you need to know. So as I mentioned, lab reports are formal papers. So they should be typed, they should be in a 12 font and Times Roman, no other fonts. That you should never use any personal pronouns. Remember, there's no use of we, us, ours, yours, I, me, etc. Any part of the lab report should not have that in it. You're going to start on your paper. Usually you're going to use some sort of Word program where in the upper left-hand corner you're going to put your name, your date, the class period that you normally have class, and then you're going to list your partner or partners. Full names, not just their first name and last initial. Okay? Then you're going to skip a space and you're going to hit center. And you're going to type out the lab number, and then under that the title of your lab. So that's the first part of the setup. Then you're going to, in the order that's listed here, have headings for your problem, background, hypotheses, experiment, data and observations, analysis, and conclusion. The headings should always be done in capital letters, all caps, a bold print, and you do not underline the headings. The headings should line up on the left-hand margin, and I recommend going into your layout or setup for your document and expanding the margins to narrow so that you have a lot of room to work with. So you're going to start off with the problem, and the problem is always going to be provided for you on the lab explanation and lab document. So you're just going to do problem in all caps, bold, and then in regular font you're going to type out the question. So it's just simply copying. Then after you're done that, you're going to skip a space and you're going to put the heading background, and again that's going to be all caps and you're going to make it bold. And then after background, this is where you're going to give a summary of the information that I've provided you in class. It's a summary of the information that is within both lab packets that you receive. And in some occasions you're going to be required to collect additional background information from a reliable source. Your background, when you do that, should apply to the problem and anything that is not common knowledge. And what that means is anything that cannot be found in your textbook or the handouts that have been provided or the information that I provide should be cited. So if you have to go to any other source other than those three, they should be cited. That even includes if you went to a friend or another teacher and they gave you information, you need to give them credit for that information. The next heading, you're going to skip a space and you're going to put the hypothesis. Again, all caps and in bold. And the hypothesis will always be put in an if-then statement. The if part should reflect your manipulated variable, what you're planning to change. And the then part should be the expected response, so your responding variable, what you expect from that. Again, always in an if-then statement, with the first part being the manipulated variable, the second part your expected response. That makes it a testable statement. And even if it is wrong, you can still test it and prove it wrong. And that's what you want to be able to do, is to prove the hypothesis right or wrong. You're going to skip a space from that, and then you're going to put the heading experiment. Again, all caps and in bold letters. The experiment should be a summary of your procedure. I don't want you to copy your procedure. I know you can copy. I want you to summarize it and put it into your own words. It can be done in sentences or it can be done in steps, whatever you prefer. It's a little bit easier in steps, but it's your preference. Make sure that you're including the information that you need to include from your experiment. Again, it's a summary. I don't want you to repeat the steps, okay? In the experiment section, this is where you also identify the manipulated variable, the responding variable, what your control is going to be, and remember your control is something that you compare the results with. It's not a controlled experiment. And if you encountered any unknowns. Sometimes you'll be given something that you don't know what it is, like you did in the organic chemistry lab, and you have to identify that unknowns were present. When you're done that, skip a space, and then you're going to put the next heading, data/observations. Again, you want this to be in all caps, and you want it to be bold. And this is going to include any of your data tables, your graphs, your charts, pictures that you've either taken or hand-drawn, written descriptions, calculations. It should be both qualitative and quantitative data when available. Most of the time it's going to be qualitative, but when quantitative is available, you need to include that quantitative. All your data tables and charts and graphs need to either be done on Word or Excel or drawn using a ruler or straight edge. Free lines are not acceptable in your data and your observations. Once you have that set up, you want to skip a space and you're going to include your analysis. And the heading analysis is all caps and done in bold. And this is going to be an interpretation of your results. It should include what the data means, how the data can be applied to what is being covered in class, and how the data can be applied to real-world experiences. How can you use this information outside of lab? It should also include any possible errors or inconsistencies that you observed. Once you're done the analysis, the next heading is conclusion. Again, conclusion should be your heading, all caps in bold, and this is going to identify if the hypothesis was supported or refuted. It's going to utilize background support and concrete observations, again qualitative and quantitative, as examples of the validity of the hypothesis. And it accounts for any errors that may have influenced the results and ties the work together. Your conclusion should never have an if-then statement in it. So you take your hypothesis out of that if-then statement and you express it in a way that either expresses support or expresses that it was refuted. If you're not sure how to write a conclusion, there's a screencast that you can watch that will take this step by step for you. Okay, so additional information. Anything that is not common knowledge needs to be cited, and you need to follow the format that you are using in English class. Remember, even if you go to an individual that is not in your lab group, you need to cite that person. If you are not using Excel, Word, or other programs to generate your graphs and your data tables, then you are to use a ruler for all straight edge work and a compass for all circular work. Lab reports are always typed. Background information can also be summarized. Spelling counts, punctuation counts, and make sure that you always proofread because I will take points off. Lab reports are generally worth 100 points. They take a lot of time to grade, so I expect you to put a lot of time and thought and effort into it. Okay, so here is an example of a lab report. Bertha Humperdinck did a lab on the 12th of September. She's period X, and her partner was Ethelbert Fingelstapper, and they were checking light intensity and photosynthesis. So they identified their problem. Does light intensity affect the rate of photosynthesis? They included their background information that you can see in here. Their hypothesis is clearly stated in an if-then statement. If the light intensity is increased for a plant, then the rate of photosynthesis will increase. They put their experiment, identifying their manipulated variable, their responding variables, their control group, and they identified that no unknowns were expected. Their experiment was to obtain 3 Elodea plants, place the plants in a sodium bicarbonate solution, place one plant in bright light, another plant in low light, and a third plant in natural room light, count the number of bubbles released from each plant over a 5-minute period, and record their results. They made a data table where you can see how many bubbles were produced in the high light, how many bubbles were produced in the room light. They should also have a data table that would include the other lights, but because of space, I did not do that for this data table. Their observations have an explanation of that data table. The above data table shows that in low light, only a few bubbles were released from the Elodea plant. Bubbles formed on the leaf of the plant but did not break free and rise to the surface. It's just a description. The plant placed in the high-intensity light had the most bubbles released. It too had bubbles forming on the leaves, but enough oxygen was produced to release the bubbles. And then the plant placed in normal light, serving as the control, appeared in much the same way as the low light and high light. Bubbles were released, but at a slower rate than that of the high intensity and more than that of the low intensity. So it's just a simple explanation of that data. Then your analysis. The data table shows that when light intensity is increased, the rate of photosynthesis increases. Oxygen is a byproduct of photosynthesis and can be detected in the water as a bubble. There were more bubbles released in the brighter light. This experiment applies to Chapter 8, Photosynthesis, and examples the light-dependent reactions. The plant in low light could not rapidly undergo photosynthesis, slowing the production of oxygen. Understanding this can be applied to helping plants grow better and more productively by knowing the intensity of light they perform the best. So then you're going to do your conclusion. Notice I do not have a restated hypothesis, I have a rephrased hypothesis. The intensity of light does affect the rate of photosynthesis. Photosynthesis is a process that requires light energy. As a result of the reactions that take place in the process, oxygen is produced and released. The plant that was placed in the low light did produce bubbles on the leaves, but did not have enough to be released at a steady rate. Only 10 bubbles were released within the 5-minute observation period. In contrast, the plant placed in the high-intensity light released 232 bubbles during the same time frame, indicating that photosynthesis was occurring more rapidly. A comparison was made with the plant at room light to ensure a change had occurred. It is noted that the plant in natural light released 115 bubbles. As light intensity increases, so does the reactions of photosynthesis, and when light is decreased, the reactions slow. Okay? That's how you write a lab report. If you have questions, see me.